Hormones obviously are important for human physiology and for the complexity which humans can attain. Hormones are not just these signals released from large endocrine glands which travel through the blood and then reach target tissues, however. They are far more diverse than that. Sometimes being signals released from one cell which affects neighboring cells, sometimes acting in chains, sometimes going from one individual, such as the mother or the cells of the oviduct of the mother, to the infant or the embryo. How did this signaling evolve? The simplest of organisms recognize signaling molecules using receptors that can be on the cell membrane, such as G protein coupled receptors, or in the cytoplasm, such as the nuclear receptors, which then go into the DNA where they activate genes. The receptors for signals which act in bacteria and eukaryotes are also the gene families which are the major receptors for hormones in organisms as complex as humans. What has occurred is there has been an amplification over time where gene duplications have taken one ancestral receptor and then made two and then four and then hundreds. Thus, the large number of hormone receptor genes in the human genome are the result of duplications of a very small number of ancestral receptor genes. Thus, at some level, the study of the evolution of the endocrine system in vertebrates could be a molecular question, asking when genes were duplicated, at what point the receptors for a certain hormone existed, at what point the enzymes to produce the hormone existed. Other aspects could be more at the cellular level. At what point did the cortex of the adrenal gland and the medulla become a solid structure where the cells which release separate signals were then organized together? At what point did the cells which release insulin and glucagon as the endocrine pancreas then become these islets inside the exocrine pancreas? And so the study of the evolution of the endocrine system could be done at a much smaller level than comparative anatomy typically studies, at a cellular level and at a genetic level.